Good afternoon or morning, everybody, depending where you are located. I am very pleased today to host this first webinar around biofilm on farm. As a product manager for animal environment product range at L'Allemand Animal Nutrition, we are developing some microbial solutions to better manage and secure the microsystems around animals that can have some negative impact sometimes, like bedding, manure, slurry, or also surfaces. And to discuss this topic, today I would like to welcome our two experts. The first is Romain Briandet. Hello, Romain. Romain is the Director of Research of the French National Institute of Agriculture. He is leading a team investigating the links between the biofilm architecture and the associated properties, and especially their impact and application in the agri-food industry. So today, Romain will give us an overview of the different mode of action involved when we talk about positive biofilm, with various examples that opened to promising application on farm. Then we will welcome Anna Rodiles. Hello, Anna. Anna is a research scientist at L'Allemand Animal Nutrition. She's a specialist in microbiota analysis using bioinformatics from gut to now environmental biofilms. And she will describe today some results directly from a farm application of positive biofilm. So during the presentation, you can ask questions in the question box of the webinar parameters, and we will dedicate some time at the end of the session to discuss it. So Romain, you can start with your presentation.
Thank you, Anna. Hello, everyone. So in this few minutes, I will talk about positive biofilm, the concept, and promising application on farm building surfaces. So as a general introduction, you probably all know that in their natural environments, bacteria mostly live associated to surface in a so-called biofilm. Once they have stick to surface, they will communicate each other, they will interact, they will produce some extracellular polymeric materials to build this kind of microbial cities, which will protect them from environmental stresses and give rise to emerging properties, including a fantastic adaptation to antimicrobial action. So if we go along the food chains, you would be able to see this spatially organized structure of bacteria or microorganisms in several places, in several locations. And for example, on inert surfaces, you can see a microscopic jungle composed of different types of organisms, matrix, and a lot of different heterogeneous material that will form those biofilms. You can also find some spatially organized community on food matrix. And for example, a microcolony of pathogens will also exhibit some features very close to the one observed in biofilm. And finally, a host can also host a biofilm on animal or human, on the skin, in the gut. The bacteria can also organize themselves as a biofilm and develop those emerging properties that can be involved in a very serious infection. To give you an idea of the diversity of biofilm structure, I took here the example of 3 strands that we isolated in a food chain. It was in a salad factory, and it was a study performed with microscopic tools that we developed in the labs that allow to see in 3D the formation of biofilm on surface. So you have, for example, on the first row, the biofilm formation of strain 1 over the days, and the biofilm were incubated at 10 degrees in this experiment. And you can see here, one bacteria is one single object that you can see at day 0 to give you a scale. And you can see that in all the situations, after 1 day of incubation at 10 degrees, the surface is almost covered by those biofilm grown from those adherent cells. Another important feature is that you can see that the biofilm structure is really specific to the strain level. Different strain will give rise to different spatial organization we cannot speak at the species level. We really need to stay at the strain level. And for example, in this experiment, you can see that strain 1 gives rise to a bubble-like structure of biofilm. Strain 2, that is forming some chains, is forming a hairy-like structure of biofilm, which is very different from the first one. And the third strain, that is producing a lot of polysaccharide, is giving rise to a sausage-like structure that are very dense and very sticky on the surface. And this spatial organization is really important at the single-scale level, because if bacteria grow as a biofilm, this will generate a heterogeneous microenvironment within the biofilm. You will have gradient of pH, of nutrients, of different stratification of bacteria. And so each cell in the biofilm will grow in a specific microenvironment that will drive diversification of cell type. It means that the same cell in the biofilm will express different genes, will have different cell behavior, and overall, this diversity will give rise to emerging community function. And this is of very strong importance regarding pathogen control in the food chain. This really changes the persistency behavior of pathogen in the food chain. One of the emergent properties of biofilm, we have seen this already, is the fantastic resistance towards the action of biocide. And so in many presentations, you can see a parallel between the microscopic architecture of the biofilm and the fortress that human activities have used in the past. And if you go through the literature, you will see that most of the bacteria, when they are forming a biofilm, they can be up to 1,000 times more tolerant to antimicrobial action than the same bacteria cultivated in a test tube as a free cell, as the so-called planktonic cells. Here, just a quantitative graph to illustrate this. On this graph, you have the log decrease of a pathogen, Listeria monocytogenes in this case, toward the time of contact with the biocide, a quaternary ammonium in this case. And the same quantity of bacteria are presented to the biocide in different forms. On the first curve here, the bacteria are exposed as a free cell in their planktonic state, so in a test tube. And you can see that in this condition, in a few seconds, all the population is eradicated. And it is what the regulation is asking to a biocide to do, just to have a big log reduction of planktonic cells. If you let those cells stick to a surface for a couple of hours, then you see that you will need not a few seconds, but something like 12 minutes to eradicate the population. But you can reach this stage where you eradicate the population. You don't reach anymore this stage when you are dealing with biofilm. In that case, even if you wait longer and if you increase the concentration of biocide, you will have a persisting subpopulation that is not eradicated by the biofilm action. And to see what happened in the structure of a biofilm, we developed some imaging tool, like here on this band, where you can see the death of a biofilm when it is exposed to a quaternary ammonium. And in that case, the dead cell appear red and the living cell appear green. And you can clearly see that over time, the death of the biofilm is slowly propagating from the edge of the cluster toward the bulk of the biofilm. And this cinetic can be very slow and can allow the bacteria to react, to adapt to sublethal concentration of the antimicrobial. So it is quite clear now that disinfection is not the golden bullet to eradicate biofilm. We can have some action, but it will not be sufficient to eradicate the biofilm. And so all the chemical molecule used in the food chain as a pesticide in the field, as disinfectant in the food processing, as a preservative in the food preservation application, or the antibiotics in the host are not totally efficient to control the pathogens, and they are for most of them, very polluting for the environment. So many labs now are trying to find microbial solutions to replace this chemical molecule in process that can be named biocontrol, bioprotection, biopreservation, probiotic. We even hear about fecal transplantation since a few years. And in all those approaches, we try to use bacteria to guide the ecology of the system to exclude pathogens from the system. Protective biofilm is one of this approach where we try to guide the ecology on the surface to favor the settlement of a protective biofilm that is going to exclude pathogen from the surface through different mechanisms that can be a spatial or nutritional competition or microbial interference through different molecular process that can interact with pathogen growth and survival. To give you some concrete example in other field, we have worked recently in the field of the French bottom mushroom sector. You have to know that in this sector, around a quarter of the mushroom production is lost because of a green mold that is named Trichoderma aggressivum, and that is very virulent towards the bottom mushroom. In France, and it is since more than 10 years now, we are using a biocontrol agent that is a strain of Bacillus thulesensis. And when you add this strain in the compost, you prevent the development of this green mold that is attacking the mushroom. You can clearly see this effect on this image. So this has been done in the Technical Center of Mushroom in France. And you can see on the first row, you have the initial system. And on the second row, you have the end of the culture with the mushroom if they could grow on the substratum. If you add artificially on the system this green mold that you can see here, you can see that at the end of the culture, you have no mushroom at all in the place of inoculation of this green mold. If you inoculated the compost with this green mold that has been protected by this strain of Bacillus, you recover almost totally the production of the mushroom. And we have some robust statistics over years of this process. And if you have a basal layer of mushroom production, you clearly see the loss of production when you inoculate it artificially with a green mold. And you can restore this level of production by protecting the compost with this biocontrol agent. And this is a biofilm story, a positive biofilm story. We develop a system to visualize this biocontrol agent in green on the compost in red. And you can see clearly some spatially organized community of the biocontrol agent forming in the compost. And we went down to gene expression level to see exactly the physiology of this biocontrol agent in the compost. And we observe that all the genes that are important for biofilm formation were strongly overexpressed in this condition. And this biofilm formation clearly give a fitness advantages to this biocontrol agent in this community, in this environment, as a spatial and nutritional level. In this model, we also detect some genes that were overexpressed and that are involved in genes coding for antagonistic molecules through interference process. So in this example, probably we have a mixture of competition and interference that are put in use to this nice bioprotective effect. But this is not always the case. And we need to understand in such a system the mechanism of the positive effect, whether it is pure competition with the pathogen or if we have specific interference mechanisms that are involved in the process. And to do this, we develop a methodology in the lab where we can study biofilm formation of strain one by one and after in combination. So, for example, here we have strain 1, strain 2, strain 3. They are labeled genetically with different color, and we can make also a B species biofilm with the two mixture of these three strains. In the first mixture, the red strain is expulse from the system after 20 hours. But this we could anticipate. We could by modeling anticipate this phenomenon just by the knowledge we have on the single strain biofilm formation modeling. So what we observe here, it's a pure competition of the strain together. The green strain is expulsing the red strain by spatial and nutritional competition. In the second case, it is different. The red strain is also excluded from the system, but this we cannot anticipate. We cannot predict this exclusion by our knowledge on single strain biofilm information. And this clearly shows that the neutrality model we can apply to this model is not validate. And there is something else occurring here, and that explains the interaction between these two species. In that case, we can talk about interference. We don't know the mechanism. It can be positive or negative, but something specific occurs between these two strains that is not that cannot be explained only by pure spatial and nutritional competition. And so here, another example where we have pure nutritional competition. This is, we have a modeling framework for this. It is called the Jameson effect. And it is typically what can occur when you have a microbiota that come in competition with a pathogen. And you have a Jameson effect when the pathogen stop growing, when the dominating microbiota reaches its maximum population density. So on this theoretical graph, you have here in blue the microbiota, that could be the positive biofilm in our case. And in the same system, you can grow the pathogen alone, in red here, or in presence of the microbiota. And when you do the mixed species community, the pathogen will stop to grow exactly when the microbiota will reach its maximum population. It's very simple. They are just sharing some common nutrients. They are not available anymore, so the pathogens stop growing. It is exactly what we observe in a real experiment with Listeria monocytogenes in the cheese field on the shelf, the wood shelf that are used for the ripening. Here you can see exactly the same type of graph, but this time they are not theoretical, they are observed data, where you can see here the growth of the microbiota on the shelf model. You can see the growth of Listeria monocytogenes alone, and the growth of Listeria monocytogenes when it is mixed with the shelf microbiota. And this is typical from a Jameson effect. The pathogen stop growing when the microbiota reaches its maximum population. You can even turn the system a bit more close to realistic condition when the pathogen is inoculated on the with the shelf microbiota after the shelf microbiota has started growth. It is more close to the reality. And in this case, you even do not see apparent growth of Listeria monocytogenes in such a system. And so based on this kind of experiments, the industrial in this area know when they are entering a new wood shelf in a room, they would wait a few days so that the shelf microbiota start to grow on the shelf. It will form a protective biofilm before the shelf are put in production and they are used in contact with the cheese. And by this, you reduce the risk of Listeria development on the shelf. And it is what we observe with wooden shelves that we take from a different producer. And so here you have the counting of Listeria monocytogenes at different time points. So we inoculated artificially in the lab in a challenge test the wood shelf with Listeria monocytogenes. When you have this native resident biofilm, you see that there is no growth of Listeria over time. And after 12 days, you have less Listeria than the initial inoculation. Just to demonstrate that it was a microbiological action, we inactivate the biofilm on the wood shelf, either thermically or by ionization. And in that case, you can see that you have a very nice growth of Listeria on the surface. because there is no nutritive competition with the pathogen that is very happy to grow in this condition. And this is a robust effect. We tested different producer, different shelf from different room, and we clearly observe the same type of effect. So really here we face a typical Jameson effect because the nutritive resource is quite limited in this condition. Then what can happen if the nutrients are not limited in the system? So it's what we tested with another system where we can grow the biofilm in a small reactor that we call a flow cell, where we can image the biofilm in real time. And we perfuse the biofilm with nutrient growth continuously. So here, as the nutrients are continuously perfused in the system, we cannot invoke anymore the Jameson effect. And here, to make a simple system, we use a lactic acid bacteria, Lactococcus lactis, that is very happy to grow in the flow cell over time when you cultivate it alone. We use Listeria monocytogenes alone, that is very happy to grow in this system when it is cultivated alone. But when you make a mixed species biofilm, once again, Listeria monocytogenes cannot grow in the multi-species biofilm. And we test several hypotheses. It is not a specific mechanism of interference here. We could explain this only by spatial competition. We use fluorescent strain of Listeria monocytogenes alone or in combination with Lactococcus lactis, and we could observe that in presence of Lactococcus lactis, the cell just lay on the surface and we are not able to grow. And by applying a spatial model of growth of this type of two species biofilm model, we could show that the spatial competition that occur between the lactic acid bacteria and the pathogen trigger nutritional competition, and this is related to the different kinetics of growth of the microorganism in this system. So spatial competition in a biofilm is really important because it will very fast trigger nutritional competition for the between the different species. And so if we now speak about biofilm formation on a building in a farm, we have a starting collaboration with L'Allemand to understand the mechanism of protective biofilm in this type of system. And there is a PhD of Virgile Guénaud that has started around 1 year ago, and that is built on 4 work packages. The first one is really a field work package where we are going to put in use cutting-edge technique to describe the community on surface of the building of this farm. And with those information, we will go after at the lab scale to identify the mechanism of pathogen exclusion by those type of biofilm by developing a model, a lab model to mimic the field condition and to study the mechanism of interaction. And after knowing the mechanism of interaction, we will develop a pipeline of analysis that allow us to select the strain with the best potential to form protective biofilm in this environment. And we will be quite sensitive also to the way of assembling this community, this protective bacteria to form this positive biofilm. And the last stage of the PhD will be to go back on the field with this new consortium to be tested in real condition with the tool developed in the program to evaluate the efficacy of this protective biofilm in real condition. And now I give the screen back to Julia to give you more information about this field experiment and the product developed by L'Allemand.
Many thanks, Romain, for this dive into the biofilm world. I hope you see my screen now.
Yes.
Perfect. So we will continue the second part by seeing concrete application of.
Julia,
Yes.
we can see your screen, but we see in presenter mode.
Okay, sorry. Just a few minutes with the technology.
Yep, that's perfect.
Here. Yes. Yes, so we will continue to see a real on-farm application. But just before going through the results with Anna, some words about the context of the positive biofilm on farm, which is linked to biosecurity. When we talk about biosecurity, we talk about some measures that can be equipment, technology, and action, and in particular some human or management behavior that will help to secure the environment for animals and also for workers and humans. There is two big types of biosecurity measures. The first one is the external biosecurity. And here the purpose is to limit the introduction of disease or bacteria inside the farm. That can be brought by water, feed, also humans, animals, not only the growing animals, but also some insects and rodents, for example. So all these entrants that can be on farm should be really managed to avoid the introduction of the disease or bacteria. The second measure that can be settled is the internal biosecurity. It means that we will here limit the spread inside the farm between batches or between the different animal stages, and also between animals and humans that are already on farm. When we look at these different measures of internal biosecurity, there is different strategies. The first one is to limit the contact between the infected animals, for example, and the healthy or sensitive animals to reduce the spread. So this is going through some management or barn design protocols that can be just to respect the density, the standard equipment, the all-in, all-out management. Another possibility as a strategy is to consolidate the animal natural defense. So here we can talk about the proper management of the feed and the water programs. And also we can see vaccination as preventive measures. And also another example is any additives or ingredients that can boost the immunity systems. And finally, the last strategy is to limit the infectious pressure. It means that we will limit the quantity of undesirable bacteria and their growth opportunity. Here we can first manage and adjust the building parameter. It can be the temperatures, the ventilation system, the bedding or litter management, and also the moisture content inside the farm. So all these parameters, they are here to manage and to make animal comfortable, so link with the welfare of animals and of course the production potential. But also by regulating them, we can more secure and limit the development of bad fermentation or bad biofilm on surfaces, for example. The other option is also to play on the hygiene rules. So it means that is regular actions that can, again, limit the infectious pressure. For example, the main one could be manure management during the batch, so to adjust the frequency of the manure removal, for example. And also between batches, we can think about cleaning and disinfection that should be done properly. And as a last step of this cleaning and disinfection, we can think about positive biofilms, as we will see. Why cleaning and disinfection is important is because it will clean the surface and make the space proper for biofilm implementation. So just a reminder of the good steps for cleaning and disinfection. The first one is to prepare the room in the barn. So after the animals are out, we will remove all the organic waste that can be in the barn. Then it will be time to do a proper detergent step. So the detergent will be soaking all the remained organic waste, and it will start to damage the matrix of the biofilms that can be remained and residual in the farm. Then by either thermic or pressure high water, we can remove the detergents and wash the building. This step also should be very well done on the water and feed system because it is complex systems where we have a high risk of remained biofilms and bacteria due to the high humidity and the nutrient availability for microorganisms. And finally, after the detergent and cleaning process, this is the time of the disinfection. So as the Romain explained, the disinfection is efficient when the biofilm is already damaged. So the disinfectant molecules can act at the close level to the bacteria and eliminate the maximum of the bacteria. Now, if we look at the microbial level and talking about quantity of bacteria during these different steps, here it's a study that have been run in a real farm, a swine farm, and the objective was to evaluate the level of bacteria during the different steps of the cleaning and disinfection. So here we can see that we have a high level of bacteria just after we remove the animal of one batch. Then the disinfection step is going to significantly reduce this level of total bacteria. So here the scale is in log of CFU as it is proper to talk when we talk about microbiology. But this decrease is representing more than 90% reduction of the total bacteria level. And now what is interesting is to look at the unoccupied period after the disinfection before we put new animal inside. We can see that if we wait 4 days, it is interesting because we still have a decrease of bacteria level. But then after these days, depending of course of the condition of the building, we can see a persistence and even a regrowth, significant regrowth of the total flora. If we look now at what can be in this total flora, and in particular undesirable bacteria we do not want in the building, like E. coli, coliforms, or Staphylococcus aureus, we can see again at the same way, the disinfection will reduce the presence of this type of bacteria, but then we can see they could persist inside the building even when it is not occupied after the disinfection. How we can place now our positive biofilm? We can place it just after the disinfection. And here you have the picture showing the concept. And again, when you look at the decrease of level of bacteria, you have the one due to the detergent step, then the disinfection step, where you reach the minimum level of bacteria pressure. And then this is where we apply our positive biofilm and positively repopulate the surfaces and the environment. What we want to do is really to reach, to make a barrier of positive biofilm and to limit the settlement of new undesirable bacteria that could appear afterwards. Now some words about the selection of the strains that can do this positive biofilm. So it is very important to have strains that form a quick and nice biofilm. So here you can see on the pictures, by using some microscopy, that we selected some strains that you can apply at the beginning. So the first picture, T0, it is a certain dosage. And then you can see that in 12 hours, this biofilm have covering most of the surface, more than 90% of the surface. As Romain explained before, also the coverage and the biofilm ability is something that almost all bacteria can do, but which is very strain dependent. So here are some pictures just to illustrate the variability of the biofilm formation by bacteria. And for example, if you want to look two very different biofilm capacity, strain 7 and strain 10, in strain 7, you can see on the first observation that the strain is doing a very large and a widespread matrix and biofilm in the first observation. But when you look at the microscopy, you can see a very three-dimension biofilm with a high shadow showing the height of the biofilm. Now, if you look at strain 10, you can see that it is forming a small biofilm surface on the observation in plates. But then when you look at the microscopy level, you can see that it's covering quite well the surface with a low level biofilm with a low height, but quite homogeneously on this picture. So after selected some strain that is forming good biofilms, we also need to be sure that it can be effective towards some undesirable bacteria. So here again, we can screen and filter our different strains by some protocols. The idea is to grow the strain we want to test as a biofilm in small coupons, for example, and then we place it in contact with undesirable bacteria. In this case, we use Staphylococcus aureus that we can manage well in the lab, and which is forming a very high biofilm challenge. So as you can see from top to bottom, here we have an increased challenge in concentration of pathogens. And on the first picture, you can see the mixed biofilm. In green, it is the positive biofilm which is marked. And in red, you can see the appearance of the pathogens more you increase the concentration. If you extract just the red part of this mixed biofilm, you can see, because we increase the concentration, that the pathogen is doing better and better as a settlement implementation in the mixed biofilm together with the positive biofilm. So now it was an example of strain 1. And if we look at another strain, you can see first in green the formation of the biofilm, which has a different look than the first strain. And if you search for the red part of the pathogens, you can see that the pathogens cannot settle inside the biofilm compared to the strain 1. So we cannot find it anymore if we use strain 2 as a protective biofilm at first. So this is how we can also check and select some efficient strain for implementation in positive biofilm on farm. And now I will let the mic to Anna that will present the concrete on-farm data we could collect in a broiler environment.
Hello again, thank you, Julia. So I am going to talk about the trial that we did. So first of all, I would like to explain the new methodology for in situ biofilm study from farms. This is a methodology that we are going to publish very soon, together with Virgil and Roman, with the INRAE people. So we can just, for definition, what is a coupon? It's a surface of a specific material where the biofilm can develop. It is very interesting because the biofilm stay intact, because we are not using an abrasive method like the soap, like physically will destroy the surface at the same time that taking the sample. So firstly, we will place the on farm after the classical cleaning and disinfection. And then, as we can see in the pictures, have attached a double-side tape under the water lines close to the animal. So this is the water line, and this is the biofilm here, the coupon with the biofilm. Then we will remove this aseptically and bring into the lab, like you can see in the other examples, with a little bit of water to don't dehydrate that. So the main trial was made in 2 livestock buildings of 800 meters square, with more than 100 coupons placed in the farms. And we took several sampling time points with replicates. As you can see the scheming here for the control and the positive biofilm, and also the pictures where they were the chicks, and here with the water line, and they were here below, they were the coupons. We can see in this video how it's applied. It's very easy, and we should we put it, as we say before, after the application building setup. And we use these to pre-colonize the surfaces, including the fresh litter before the entrance of the animals. In the trial, we have some different time points, like the scheme. We can say that the day -1, it was the cleaning and disinfection step. Then in the day 0, we applied the positive control, and then we took samples of the coupon on the day 3, on the day 6, on the day 10 and 18, with the entries of the animals at day 4. So from day 4, all the coupon sampling were including the animals. Once that we get the coupons, we analyze them with the metagenomics, with high-throughput sequencing, and as well with a bacterial challenge with a non-pathogen. So a little bit, in short, some metagenomics explanation of amplicon sequencing of 16 rRNA. Is based on gene amplification and on gene marker methods. This allows us to study the microbial community composition, microbial networks. For example, it is a tool to understand the microbiota. There are 3 main components we have to study. The relative abundance, which is the proportion of a given microbial species in a microbiota sample or in a group of samples. Then we also have the alpha diversity, which is the diversity within one of the microbiota sample. In this case, if we count the number of different species or ASVs, it will be richness. And if we take into account the relative proportion in between the other bacteria inside of the same sample, it will be evenness. For the beta diversity study the microbiota structure. So study the similarity or dissimilarity in the microbiota composition between sample, and it's a multivariate approach. So coming back to our trial, the relative abundant results we can see here that there is an increase of the Lactobacilli family, and also a decrease of Enterococcaceae family and Enterobacteriaceae family during the first week and overall of the breeding period. So here we have the plot of overall. We have almost 40% of Lactobacilliaceae in the positive biofilm, but not in the control. It's not reaching. It's reaching a little bit more than 10, and the other 2 families really get much more than in the positive biofilm. If we look very interestingly to how the animals change this microbiota composition of the biofilm or abundance of the biofilm, we can clearly see how before the animal's entry, we reach almost 80%, well, more than 80% of Lactobacilli when we apply the positive biofilm. But this is not the case in the control again. And we are also able to decrease numerically the Enterobacteriaceae. As you know, there are typical pathogens like Salmonella or Enterobacter or E. coli as well, part of this family. And we can also, after when the animals enter, we see a very big increase of these 2 families. Enterococcaceae in this case was mainly coming from Enterococcus secorum, which is a very well-known pathogen of in chickens. But for the positive biofilm, we have a significantly higher increase of lactobacilli and a smaller increase of Enterococcaceae at the day 6. When we go to the alpha diversity, we can see how the biofilm modulates, the positive biofilm modulates the microbiota composition across the experimental period by stabilizing the Shannon diversity over the time. How we can see this is like at the beginning, when the animals enter, the positive control reach a smaller Shannon than in control with significant differences. Then biofilm goes up and control drops very quickly. And we have to remember on the slide before that that was due to the higher abundance of Enterococcaceae and Enterobacteriaceae families in the control group, which is not happening in the positive biofilm, that little by little with the time is allowing the animals, well, we don't know the animal, but at least the biofilm is stabilizing in the animal's environment, which is very important. And we also will avoid the last goes up in diversity at the end that is happening with the control. When we talk about beta diversity or bacterial community structure, it's important to explain in a very simple example what it is. So for example, we have a group 1 that have 3 bacteria, 3 green bacteria, 3 pink and 1 orange, 1 individual here, also the second individual and the third individual. We could say that the microbiota composition or structure is very similar about the samples, for even they are even equal. But in the opposite, in the group 2, we have a different composition of the red, sorry, of the green, also the orange and the pink. So we can also say that the composition of the microbiota is very different among the samples. So in this case, we will have a high beta diversity distance. So this is mean that they will far away plot in this example, for example, that we have here. So these two will be very far away from each other. But looking at this specific paper with the Sang et al. In 2018, we see how the ileum clusters or plot together with all the samples and very separated from the cecum and colon, which is obvious because we know that in the gastrointestinal tract, the ileum is very separated physically from the cecum and the colon. So the communities that are typical in the ileum will be very different from the colon. Mainly in the colon, they will be with no any kind of oxygen present. So if we come back to our trial, the chicken trial, we can see in this case, this beta diversity plot, how the positive biofilm modulated the microbial composition along the trial when we compare with the control group. This is the day 3 with differences with the control, also the day 6, the day 10, and the day 18. However, the non-treated group, so the control, did not change much over the period, so it's staying in the same place of the plot. So we can say that in this trial, positive biofilm were able to influence the microbial environment for 18 days. Then, in the bacterial challenge test, we tested with Staphylococcus aureus inoculation on the coupons from the farm, and as well from sterile coupons to see, as well to use it as a positive control. So we have this Staphylococcus culture. We pour it here onto the media. Then we place one or another, so coupon the day from the day 18 or an sterile coupon for the adhesion during 1 hour or so. Then we reseed to remove all the excess from not adhesive bacteria. And then we place it for the incubation of 48 hours with a wet tissue to don't lose, humidity. And then we will look at it under the scan microscope with fluorescence, as we have been mentioning before. In this case, we have this Staphylococcus aureus alone on the sterile coupons, so we can see more red. And as well here, that is reaching the higher amount of fluorescent biovolume. Then we also have the control biofilm from the farm, which have a little red. And finally, the positive biofilm from the farm with have mostly the green bacteria. So we can know for this plot that the natural biofilm protects the surfaces from Staphylococcus aureus, even if we don't apply a specific positive biofilm, but the positive biofilm contributes to an extra additional protection because we have significant differences on the role of this pathogen in comparison with the control, and also even more, obviously, with the positive Staphylococcus aureus. So to conclude, surfaces on farms are covered by natural biofilms. It is important to study in situ biofilm evolution in complex environments with a new methodology and biotechnology tools, and with a rationality to select the strengths. Positive biofilm is a promising new concept to better understand the influence of the microbial environments in the farms, and it is necessary to set some guidelines for microbial ecology of surfaces to secure animals' production. And obviously, there is always a less risk of health issues by preventing the microbial pressure of undesired bacteria. Thank you very much. Any questions?
Thank you very much, Anna. Now we will have some minutes dedicated to the questions. So we will go through some of them. Don't hesitate to use the question box there. First one, maybe through following Anna's presentation, will be to know what bacteria was in the farm controlled biofilm. Do we know a little more?
Well, as we can see in the alpha diversity, it was composed by another families. There were most of them also present in the biofilm, but in higher amount and with the higher diversity. So they are typical bacteria from the chicken farm.
Perfect. Now, one question maybe for Romain. What is the link between the three-dimension structure of the biofilm and the surface covering?
So the importance of the spatial organization of the candidate for the biofilm.
Yes.
Yes, that will be one key question of the PhD we start, and that will probably be some important selection criterion for the next generation cocktail of strain that can form this biofilm. And nowadays we don't have a clear answer to this question. We know this spatial organization is really dominating factor towards the colonization of pathogens, but now we need to analyze it in detail with the high throughput tools that we develop and put in use in the project, and we hope to have the answer in probably 1 year to this question.
One question is if we plan to run some experiments to measure the actual health outcomes of the animals in control building versus inoculated building. So yes, it is the next step. We already have some positive testimonial from some farm application in the field by, for example, measuring some of the health issues. It can be diarrhea, or it could be also the amount of medication used in the different batches. We could see some impact on mortality, for example. So any kind of health-related parameters that could be affected by the environment could be improved by this positive biofilm. Another question, maybe for Romain. We learned a lot about the quorum sensing related to biofilm. How this can be involved in positive biofilm?
Yes, so quorum sensing is a general trend of bacteria when they are attached to a surface. So each bacteria can secrete a molecule that it is also able to detect. And above a certain threshold, it will activate a profound reprogrammation of gene expression. And this can be a target for alternative approach where we are going to to interfere with this communication system to prevent the maturation of the biofilm from the pathogenic side. So yes, it's a kind of interference mechanism where here we are trying to modulate the communication between the bacteria to prevent the most unwanted form of the biofilm.
Thank you. One question for Anna, why we did not sample after day 18?
Well, it is what you say at the beginning. We were testing at the beginning how it was for the first 18 days. So that we will do in the future what is happening, and that's for sure is part of Virgil PSD. So we're working on that.
Yes, thanks. The first step of following the disinfection and the entrance of animals is very sensitive period, so it is what we wanted to look at in deep at the beginning. Maybe a question for both, if you have some experience, if we can compare the positive biofilm to the positive effect of probiotics on the colonization of the digestive tract. I don't know who want to give his opinion.
We can make a parallel for some mode of action of the probiotics. The probiotics have several action in the digestive tract. They can interfere with pathogen colonization, and on this aspect, yes, we can have a similar mechanism in protective biofilm. But probiotics also have a immunostimulation effect that is a deep interaction with the host that we are not considering here because we are on inert surface. But for the barrier to the pathogen development, yes, we want to guide the ecology of the system, and we can invoke similar mechanisms.
Thank you, Romain. I think we are at the end of the session now. So, of course, if you have more questions, don't hesitate to send us by email. We will come back to you for the questions we could not answer due to the time. Thanks a lot, both Romain and Anna, for this presentation. And I hope you all enjoyed this webinar. Have a good day and see you soon.
Thank you.
Bye.
Bye.